Yeah, it recorded a different part. Okay. So sorry for this delay. I had some technical difficulties, but now we're ready here and Sharon is going to give her talk. So today we have Sharon Ruiz López, and she's talking about microbial ecology of extreme environments, analysis of open air and indoor spent nuclear fuel storage ponds at Sellafield in the UK. Sharon did her undergraduate studies on biochemical engineering and then got a master's degree on biotechnology and bioengineering at the National Polytechnic Institute in Mexico. She studied the bioremediation process of areas polluted with pesticides. Then she moved to the UK, where last year she earned a PhD in earth sciences at the University of Manchester. Her project focused on studying the microbial ecology of radioactive sites by using classic microbiology and whole genome sequencing techniques. To have a better understanding of the microbial adaptation processes and the interactions with the environment, including the identification of virus-host interactions and its influence on key metabolic processes. Recently, she started a postdoc position at the Manchester Metropolitan University in Manchester in the Department of Environmental Microbiology. Her area of research is the microbial ecology of extreme environments. Her research is centered on acid main drainage arising from an abandoned copper mine, particularly the microbial mediated remediation processes occurring in a freshwater wetland. The aim is to understand the functional and ecological niches of microorganisms present along this stress gradient. So yeah, Sharon, please just take it from here.
Okay, thank you again. Thank you for the introduction, and thanks to the ISME Early Career Scientist Committee for inviting me and for organizing this series of seminars. I have really enjoyed watching some of them, so thank you again. And so, well, we'll start over with my presentation, and I'm going to share a screen right now. That's it. Okay, good. All right, so here we go again. So I want to talk to you a little bit of the work that I have done during my PhD and that I intend to do right now in my postdoc. The first part is the project that I did during my PhD on the University of Manchester in a collaboration with Sellafield Limited. And the second part is the project that I'm currently working on at the Manchester Metropolitan University, also here in Manchester. So first of all, I work with extremophiles and organisms, and also I work with extreme environments. So first of all, we define an extreme environment as those environments where at least one physical or chemical property is outside the normal range for human survival, such as radiation, temperature, pressure, pH, among others. And one of the most widely studied extreme environments is the hot spring in Yellowstone in the USA. And the reasons why we studied extreme environments is because we aim to have a better understanding of the physical parameters that define life and how life has evolved over time. Also, we want to define the limits of life, so and physically and geographically. So we want to know where is the limit, where is the limit where we can find life on Earth. And also the final reason is because we want to provide an insight into life and how it's able to survive, to adapt to different environments, and if it can be a help to have an idea, to get an idea of the survival in different planetary bodies. This is an area called astrobiology, but there's something different. So I like this picture. I found it in this paper. It's very good because it shows the extreme environments on Earth. And as we can see, most of them are natural and extreme environments, such as polar regions, the deep sea, hot springs, deserts. And we also have some human-made extreme environments, such as those created by nuclear activities or mining activities. And we want to study these places because we know microbes react to a sudden change in their environment and by the expression or repression of certain genes. So we want to know what is occurring on this kind of environments, and that's the whole point of extremophiles and microbial ecology of extreme environments. So for the first part of my project, I want to talk to you about the human-made extreme environment, which is spent nuclear fuel storage ponds at Sellafield here in the United Kingdom. This is a project where I work with this, obviously with this company and the university. So first of all, here's the nuclear fuel cycle, which represents the progression of nuclear fuel from creation to disposal. It includes different steps from mining and mining, conversion, enrichment, the power generation, the spent fuel storage, the reprocessing, and final disposal. And here, the crucial step is the spent fuel storage, because after the production of power, the generation of power, we have all the fuel that has to be treated or preserved here in this storage place before sent to whether reprocessing or final disposal. So that's why this step is crucial, because it defines if the material still works or it doesn't anymore. So we studied this part. And well, what we know is that the fuel, once the fuel has been used, it's called spent fuel, and it's stored in these large cans that are recovered with concrete to protect against radiation and to protect also the workers in the areas. And the material, the spent fuel storage cans can be stored whether in fuel ponds, with water, or in dry casks. Most of the material is stored around the world is stored in fuel ponds, which are literally huge pools that are filled with an enormous amount of water. And the water has the advantages that it provides a shield against radiation, and it also helps to cool down the spent fuel. And sometimes they use different reagents added to the water. It depends on the plant. And now, let's talk about Sellafield. Well, Sellafield is a nuclear reprocessing, nuclear waste storage, and nuclear decommissioning facility. It's located on the west coast of England, here in Cumbria, in this part, and it's a massive site. Here we can see the picture below. And what we know is that it's Europe's largest nuclear site, and includes more than 200 nuclear facilities, and includes more than 1,000 buildings. So it's like, it's huge. And it also receives spent fuel from different stations across the UK. So it has the huge responsibility to handle all the material from different sites. And when spent fuel arrives to Sellafield, as I mentioned before, it's kept on the water in these storage ponds, where the water helps to cool down the spent fuel, and also it has to stay in this pools for a period of time of at least 100 days to help the radioactivity to decay. And for these ponds are filled with demineralized water, so there are no carbon sources, and also the sodium hydroxide is added to minimize the corrosion. And the ponds where the spent fuel is stored can be whether open air systems like this one and in this picture, or can be indoor systems like this one below. So these conditions create an extreme environment because of the addition of sodium hydroxide, the environment is hyperalkaline, where the pH is around 11.5 or 11.6. And it's also an oligotrophic environment because there are no carbon sources. And of course, it's radioactive because of the material that is stored there. And also, in the case for the indoor system, there are no sunlight sources. So it's a poly-extremophile environment. And despite these conditions, we know that microbes can survive and live in this kind of environments. So we decided to study these places. We wanted to know what is occurring there. And we formulated these research questions. The first one, if there are organisms surviving in this kind of environment, who is there? What are they doing to survive and colonize the pond? And if they can influence the spent fuel storage system. Because we know when microbes grow, they create biofilms, and the biofilms can affect visibility, which also increases the cost of operation and affects the plant, obviously. And also bacteria can promote the corrosion through a process called microbiologically induced corrosion. And also bacteria can interact with radionuclides and by different mechanisms such as bioaccumulation, biosorption, biomineralization, among others. So I wanted to know what is occurring in there and how we can be controlled, or if it affects the plant or the storage systems. So we studied two different storage systems, an open air, which is the one here in orange, and an indoor system, which is this one in blue. For the indoor system, we call it indoor pond or IMP. It's this pond where the material is stored, and it includes a feeding tank area where the water is purged. So in this part, there are no direct contact with the material, just the water is purged. But in the material is stored in this part. And for the outdoor system is the first generation magnet storage pond, this one, or FGMSP, this one, and it includes an attached auxiliary pond. And the material is stored here. It's stored in both systems, but let's say the main pond is this one, FGMSP. And these ponds are hierarchically linked, not disclosed, but they are linked. So we wanted to know if there are any the microbial diversity was different or contrasting between these sites, and if it's similar, why is it similar, and what can you tell us from the activities occurring in there? So now to take the samples, to analyze this place, it was very difficult and highly risky because we know we are working with radioactive samples. So we didn't want to take any risk, and we had to follow the protocols from Sellafield and also from the nuclear organizations here in the UK. So at the end, we decided to work only from DNA. We decided to take the samples from here in these areas, but the DNA extraction was carried on Sellafield, and we only transported the DNA to the University of Manchester. So at the end of the day, we only work with DNA. So we decided to work to split the work in two parts. The first one was to assess the taxonomy diversity. And for this part, we did amplification and sequencing of the 16S RNA gene. And for the functional diversity, we had a chance to do whole genome sequencing and to do the assembly and functional assignment. But initially, we decided to go to use the MG-RAS server to have an initial insight into the metabolic activities that might be occurring in this pond, in this system ponds. So for the first part, for taxonomic diversity, let me see, so the first two bars represent the feeding tank area, which is this part here, where the water is purged. The middle bars represent the indoor system, this one, and the final four bars represent the open air systems. What we found is the microbial diversity was consistent. For example, from the indoor pond, when we took the samples at different times, we found the same diversity, and also for the other systems. And we found also that organisms affiliated to cyanobacteria were detected exclusively on the open air ponds, in this one. And when we look at genus level, we found very interesting information. We found that this pink bar, this one, it's very abundant, and it represents organisms affiliated to genus Hydrogenophaga from phylum Proteobacteria, and this bacteria is responsible for metabolizing hydrogen. And we also found photosynthetic organisms. Oh, sorry. Photosynthetic organisms only on the open air systems, such as this one. So what we have here, we had the theory that since we found organisms affiliated to Hydrogenophaga, possibly the metabolism of hydrogen could be the main energy source in the indoor systems. And since we found organisms related to photosynthetic activities, maybe the photosynthesis could be the main energy source in the open air systems. That was a theory. We didn't know for sure, but we know that there were no other energy sources such as carbon or nitrogen. So that was our first theory. So we decided to look at the functional diversity. And initially we have all this data, and it didn't tell us much. So it was just like a regular graph. So we decided to analyze data, and we standardized it, and we created a heat map. Here we have it now. And it was very interesting that what we found here is that on the feeding tank area, which is again like the initial part, we found a high relative abundance of genes related to motility, to stress response, to potassium metabolism, even to membrane transport. And in the indoor pond, which is where the material is stored burned in indoors, we found high relative abundance of genes related to respiration, sorry, here in the middle, and DNA metabolism. And for the open air systems, we found a high relative abundance of genes related to photosynthesis. So now we have more evidence saying that possibly, yeah, hydrogen could be the main energy source in the indoor systems, whereas photosynthesis could be the main energy source for the open air systems. And we decided to look at a higher level, a different level, also provided by MG-RAS. And we found that on the feeding tank area, the initial part here, we found bacterial responses to osmotic stress. We found abundance of ABC transporters and and also the control of intracellular potassium and sodium ions. So with this, we can have an idea of what is occurring here. So possibly bacteria, since they received the first osmotic shock, they have to control the internal pH, and they do it by using ABC transporters or channels to control the potassium ions. Oh, sorry. And now, when we look at further higher level of analysis for genes related to respiration, what we found is that we found the abundance of genes related to hydrogenases, this one yellow here. And we also found the relative abundance of genes related to hydrogenase maturation activities here, this green bar. And these were more abundant on the indoor systems, which is the bars here in the middle. And also we had the chance to look into to further level to try to find the link between the genes and where they come from. Sorry about that. So where the genes come from, and we were able to track it because of MGRAS data allow us to do it. And we found that the gene related to hydrogenases came from organisms related to Burkholdeриalis, to order Burkholdeриalis. And also the genes related to hydrogenase maturation processes were also related to Burkholdeриalis activities, Burkholdeриalis order. And we know Hydrogenophaga belongs to this order, so maybe we have more evidence supporting our theory that hydrogen metabolism is the main energy source in the indoor systems. And something interesting we found here in genes related to photosynthesis. What we found here is the abundance of genes related to photosystem 1 and 2. Those were detected exclusively on the outdoor, on the open air systems, as expected. And we found that those were related to organisms affiliated to Crocoales, which are cyanobacteria. So yeah, so again, apparently photosynthesis might be the main energy source for the open air systems. And we also took a look into the DNA metabolism, to genes related to DNA metabolism. And we found a high abundance of genes related to restriction and modification systems and to CRISPR systems. And those were also related to the order Burkholdeриalis. We need to look further into this. We had just like an initial insight, but what we know is that we have other adaptation mechanisms besides the energy metabolization. So in summary, what we found is we found different microbial adaptations. First of all, apparently the metabolism of hydrogen might be the main energy source in the indoor system. And also we found that photosynthesis might be the main source of energy in the open air systems. And the hydrogen comes from the water radiolysis, which is a process where the water is exposed to radioactivity and it breaks down and releases hydrogen. So maybe that's why we have hydrogen, and that's how microbes are metabolizing it. And also we found other adaptations like stress responses to hyperalkalinity, and we also found DNA responses to external stimuli such as DNA repair and CRISPR systems. We also found that Hydrogenophaga, despite is defined as an alkali-tolerant organism, the optimal pH that we found in some papers is 7. And also talking about pH, the most abundant organisms in the whole system, the optimal pH is around 7. Only for Selenomonas and Cyanobium and Bellela, the pH is higher. But still, we found these organisms surviving and thriving on pH and hyperalkaline at 11.5, 11.6. So definitely, we know now that these organisms can definitely survive and adapt and thrive in this kind of hyperalkalinity environments. And also we found that eukaryotic organisms were detected exclusively on the open air systems, possibly because of the exposure of sunlight. And shockingly, we didn't find any organisms affiliated to archaea. We actually, well, personally, I expected to find organisms affiliated to this phylum, but we didn't find any of them. And even when we did the whole genome sequencing, I think in just one sample we found archaea, but it represented less than 1%. So basically, there are no archaea organisms there. And also, we found organisms that were not previously identified. We compare with different with storage systems, and we didn't find any of the organisms. The microbial diversity was contrasting. And also, something very interesting in this project is that we found a high percentage of unidentified organisms. For some samples were above 30%, which is super high. So one of the suggestion was we maybe need to improve our our techniques. Maybe we can carry on manual metagenomics, with which actually we're trying. And also the other explanation is that maybe we have a new species and that we are discovering. We don't know yet. And also, we need to find more evidence. We found this CRISPR system, but it's more like bacterial CRISPR. I'm not sure about this part, but since we found these genes, it's necessary to find more interaction between the CRISPR and CRISPR host interactions. That's what we're currently doing, actually. And also, this is very, very important. We found genes related to some activities, and these were relatively abundant. But we don't know for sure if the activities are actually occurring because we only look for relative abundance of genes. We need to carry on some transcriptomics experiments to confirm that these activities are actually occurring. And also we have potential applications for final disposal. This is just an initial suggestion. We need to work further in this. But the idea for final disposal is to keep all the spent fuel to bury it underground, like in tombs, like very, very deep. And the idea is to put all the material there, so it cannot affect other environments. But one of the biggest problems is that the interaction between the material, the material with the concrete or with the material that is recovering. So and it releases it releases hydrogen. The hydrogen at the same time can produce leaks here on the ground, and hence can propitiate the liberation of radionuclides, which can affect different environments. So if we can use these organisms that we detected as a biobarrier, it could be helpful for this kind of systems. It's just an idea. We still need to look further on this. But so far, that's what we have with this human-made environment. And now for the second part, I want to talk to you about a natural extreme environment. I quote it natural because it's not really natural. It's affected by human activities. So this project is located on, well, we will be working with a place in Wales, here in the UK, and we'll be working with this Paris Mountain, which is a place here in Wales. And well, it's a result of mining activities. And by the end of the 18th century, it was the largest producer of copper in the world. And it is estimated that more than 130,000 tons of copper were extracted from this site. So, and also after more than 100 years later, the mining operation ceased. There are still high releases of iron, copper, and zinc. So this is like a very polluted area. And the biggest concern here is the acid mine drainage, which gives this color. So what is acid mine drainage? Well, acid mine drainage, we know that all the metal mines with the target ore, such as gold, silver, copper, among others, it's often rich in sulfide minerals, which also it's called pyrite. This one, when the pyrite is in contact with oxygen and water, it produces sulfuric acid and sulfates, which can affect the earth. This is actually the one that provides the red color that we saw in the picture. And it also creates sulfuric acid. This acid can dissolve other harmful material, other harmful metals, such as metalloids, such as arsenic and copper, among others. And it can affect other environments. So it affects because it creates an acidic environment where the pH is it's 4 or even lower, but also we have found that the mechanisms to mitigate the AMD impact includes the construction of wetlands, the anoxic limestone drains, the addition of chemicals to increase the pH, among others. So what we have here is the wetlands. We focus on the wetland systems. The wetlands are sorry. The wetlands are these water bodies here that can provide, and this can provide an effective long-term remediation of contamination from this kind of sites. And also what differentiates the wetlands from other aquatic environments is the presence of plants of aquatic vegetation, such as this one. And also these plants are well known that they can absorb high levels of metals. So I don't speak Welsh. If someone can speak Welsh, can you just please tell me how you pronounce this one? Well, this is the name of the wetland here in Paris Mountain. And studies have revealed that this wetland is highly efficient for the removal of contamination by AMD by a combination of methods such as metal sequestration, metal sedimentation, alkalinization, and others. So we know all of this. So what is next? Well, so far we know there have been 3 projects on my group that have worked with this with this wetland. They have looked for they have analyzed the microbial diversity. They have also analyzed some of the most abundant genes, so they have an idea of the metabolic pathways that are occurring in this wetland. So what they have done is that they have analyzed, this is the Paris Mountain, they have analyzed this area here, this part is the wetland here, and they also have analyzed the river on a side, and they have compared the microbial diversity and some metabolic activities. About the microbial diversity, what they have found is that, sorry, the diversity in the wetland, as we saw here in this part, the diversity is much higher in this one. It's 1 and it's 2 and it's 3, and also they found that that pH is a determinant factor that influences diversity of microbial communities. The pH varies among the wetland, and in the wetland the pH is higher compared to the AMD polluted areas in the river. So the pH is an important factor, and also the conductivity and metal concentration is an important factor that those are important factors that can influence the microbial diversity. And they also found that the most abundant organisms here, Methylophales and Gallionella, are associated to iron oxidation. So that's what we know about the diversity in this area. And they also analyze some of the anoxic areas. They use the sampling cores, which is like tubes that they can go like deeper to analyze the anoxic zones. And they found a higher concentration of iron and iron oxidation genes on the surface here. And they also found, they concluded that the bacterial metabolism enhances the iron and sulfur transformation that leads to the rapid metal attenuation. So definitely there's a lot of metabolic activities occurring in this area, and those have been explored already. So we can have an idea of the metabolic activities, the metabolism of iron and sulfur occurring here. So with this, what is next? What is next is that they have studied the river here, the river, which I can pronounce the name, sorry about that, here, and they also have studied the wetland in this part. So what is next is that we want to track seasonal changes. The samples were taken during autumn, so we want to take samples in summer and also during spring, because the amount of water can change. So we want to track these changes. And also we want to track other genes for adaptation responses. So far we know the metabolic adaptations for energy sources. We want to track other genes such as the oxidative stress, the potassium and sodium metabolism, maybe that can also tell us what is occurring there. So maybe as we saw previously in the other project, the potassium metabolism was a key gene, a key factor that helps to control the inner pH. So maybe it can also help in this part. So we want to also analyze that part. And we also want to target other genes involved in other metabolism, not only sulfur and copper, as we already know. We want to track maybe hydrogen, carbon, nitrogen, any other gene, any other thing that can tell us more about the microbial diversity and microbial metabolism. We also want to analyze additional areas at different depths. We want to analyze, besides the river and the wetland, we want to analyze the ponds here. and in the mountain. And something interesting about this, about the ponds here in Paris Mountain, is that the pH is different in the highest part of the mountain compared to the lowest part. So that's what we want to know, like what is occurring in there. And of course, we want to understand the impacts of AMD on the microbial ecology and evolution. And we want to identify organisms that may have bioremediation properties and potential. And we want to use complementary omics. We want to use transcriptomics and metagenomics to have, or even proteomics if possible, to understand more about the adaptation systems. And we also want to study further eukaryotic diversity. We want to study if there's a possibility to find maybe fungi organisms or the interaction between plants and microbes. We want to know a little bit more. We want to know more about these systems. So in conclusion, we know that microbes display a range of adaptation strategies depending on interaction with the surrounding environment, and the approaches to study these environments and these interactions should be inclusive. So definitely we need to include isolation techniques, microbiology, classic techniques, cultivation, and obviously omics. And sadly, for my first project, we couldn't do any cultivation, but ideally we should be able to do all of this, and hopefully we can do it for this, at least for the next project. So I just want to thank for the ISME committee for this invitation, to my GeoMicro group, to John Lloyd, Lynn Foster, Chris Budman, Hu Kyung Song, Sophie Nixon, for my current group at the Manchester Metropolitan University, to Andy Dean, Damien Rivet, Richard Preciosi, to my sponsors, the University of Manchester, Sellafield, the Manchester Metropolitan University. And if you have any comments or questions, I'm more than happy to discuss. Thank you very much.
Well, thank you, Sharon. That was really interesting. And thanks for keeping the nerves after the second start.
No worries, it's fine.
You were good. So I have a few questions to start with. So I guess in the beginning, I was a little bit nervous and I didn't really hear from you, or I kind of missed it, what these plants are actually used for.
Well, for the first project, or for the
exactly for this plant in Sellafield?
Well, we wanted to know the microbial diversity, and it was affecting somehow the storage of spent fuel. And we concluded that there are no affectations, since we have microbial diversity and we have microbes surviving and thriving there, but they do not seem to make a difference, a huge difference or a huge impact on the material. So the plant wanted to know if they had to increase maybe the amount of reagents, or use a different reagent to control the microbial diversity and the microorganisms there. But we concluded that it may be not necessary. I mean, the organisms are perfectly fine there, and they are metabolizing the hydrogen that could be released there.
Okay. Is this a radioactive environment?
Yeah.
Okay. So that might explain, because then in the results, you actually also show that there you found, I think, some enrichment of like DNA repair genes.
Yeah.
I was just wondering if that maybe had to do somehow with the exposure to radiation, that they might have more.
Yeah, that's also one of our theories. That's why we look for genes related to DNA repair, because we know that's one of the strategies for interaction with radioactivity. So yeah, that's why that's why we found these mechanisms.
And also, I remember there were some studies where, when they send like spacecraft in outer space, they always have to clean them first with all types of methods. And I know they also try to like radiate them away. And I was just wondering if you maybe found some of the survivors that we actually know made it into space, if those are maybe the same taxa that you find as the survivors in the plant.
Wow, I'm not sure about it, to be honest. I mean, I think the diversity is, to be honest, the diversity that I found in this ponds, it's not what I was expecting, if we can say that, because compared with other sites, I was expecting maybe Archaeas or the Inococcus, different species. What we found here is completely different. So I'm not sure if these organisms can survive to other radioactive environments, because they are like very specific for this site. So yeah, I'm not sure about that.
Yeah. And then I was wondering, you said in the beginning that the first project was a collaboration between Manchester University and a company.
Yeah. So
And I was just wondering what is like the interest in the company in this kind of research?
well, currently, Sellafield and other companies here in the UK, they are looking for the best strategy for final disposal of the nuclear waste, because nuclear waste, despite the amount is less compared to other energy sources, is still very dangerous, and it can affect different environments, and their activity is something very risky. So they are looking for the best strategies to find a safe final disposal. So that's why we are looking for this. My project was only focused on microbial diversity, but they have many other projects, and they support many other PhD students and postdocs, and they're looking for different strategies and they want to look for interactions between radionuclides and organisms, and maybe the interactions between the material that reinforces the cans, such as concrete. So they want to know that. They want to know like the best way to dispose safely the material. So that's why they're looking for all the possibilities.
Okay. And we have a question from the audience. They're asking, why are you interested in the CRISPR and restriction modification systems in the radioactive sites? Do you expect that they play a different or special role in this extreme environment?
Well, in this case, I think, as we talked about this before, I think I look for the systems because we have a radioactive environment, and we know that one of the strategies to adaptation for this kind of environment is by the DNA repair. So that's why we're looking that we were looking for these genes. For the CRISPR, actually, it was just like a coincidence. We were looking for the relative abundance of genes, and we found the relative abundance of genes related to CRISPR that was higher. So that's why we were just interested. I'm not sure if it will make like a huge impact, because the relative abundance was very low compared to other functional genes. But I think it was interesting to take a look on it. Yeah. So, and also the next part of the project, well, I didn't show it here, but I'm trying to also find any other interactions between CRISPR and their host. It's just to have an idea of what is occurring. I don't think it will make a huge difference, but I think it's a good start to have like a starting point. So now we can know more about the interactions and the behavior of microbes and CRISPR systems.
Mm-hmm. And for the second project, did you also look at whole metagenomes, or was this based on taxonomy?
This is only based on amplification of the 16S amplicon. It was just so far. But yeah, I want to do whole genome sequencing, but we haven't done it yet. But that's the plan. We want to do it.
Yeah, because I was just thinking, if you showed in one of the last slides, you would show that map with that environmental gradient where you have several spots along the gradient. And
Yeah.
maybe it would be interesting to see some corresponding gradient in gene functions. And then you could even check whether these gene functions are found in the same taxa, or maybe depending on the environment, you might find the same genes, but then in different taxa that are able to survive.
Yeah, definitely. I think that would be very, very interesting to find to compare the diversity and the functionality between the depths. I think that's also very interesting, because the amount of oxygen will change. So yeah, I think that's very, very important.
Yeah. So I think these were all questions for today. Thank you so much for talking to us and taking the time and your
Thank you.
patience.
It's
It was
okay.
a
Thank
very
you very
nice
much
talk.
for inviting me.
Thanks.
Thank you. See